Audio Jungle.
Hello, and welcome to our 10x-pert workshop for the single-cell sample prep techniques and the best practices session. I'm Shauna. I'm the 10x community manager, and I'm going to be your moderator for today. So before we get started, let's go over some quick housekeeping items. On the next slide, we'll go over so we're going to start with a presentation by our 10 experts, followed by an open Q&A session. So we're recording this webinar, and after the session, we'll make it available on the 10x community. So by default, all the attendees are on mute. To ask a question, we ask that you please type it in the Q&A box on the right side of the screen. If you don't see the box, it may be minimized. So all you have to do is click on the little Q&A icon, and it will open up the box for you, and you can type all of your questions for the 10 experts in the Q&A box. Also, we're excited by the large amount of interest in the session, and we'll try to get to as many questions as we can. And also, if you're interested in tweeting during the session, we ask that you use the hashtag 10expert. All right, now let's meet your 10 experts for today. So our host today is Jill Hirschleb. She's the sample prep group leader here at 10x. Jill has a PhD in cellular and molecular biology and has spent the last 15 years working on technology development for high-resolution genome analysis, including optical mapping, long-read sequencing, and precision molecular counting. A fun fact about Jill is she's been working with high molecular weight DNA since 2003. And she'll be joined by fellow 10 expert Jessica Terry. Jess has a PhD in analytical chemistry and has worked for the last 4 years on product development and systems integration at 10x. She is currently a product development core team leader and a resident single-cell sample prep expert. A fun fact about Jess is that she's very closely involved in the development of the chromium single-cell 3' solution, from feasibility all the way to the solution you see today. So without further ado, I'm going to hand it over to Jill so she can get started with her presentation.
Hi, everyone. Jill, thanks for joining us today. So an agenda for today's workshop will be an introduction for our single-cell sample prep resources from 10x, as well as some general sample preparation tips, and followed by a spotlight on 2 of our newest demonstrated protocols from 10x. Those are isolation of nuclei for single-cell sequencing and removal of dead cells from single-cell suspension. And we'll leave a lot of time for Q&A afterwards, so please use the function to submit your questions. Sample preparation for single-cell sequencing is critical and challenging, and that's probably why you're joining us in this workshop today. And when we say sample preparation, we mean all the steps involved, from starting with a biological sample and ending up with a suspension of cells ready for adding to our single-cell 3' V2 solution, so clean and at the right concentration. And because sample prep is challenging and complex, at 10x we develop a lot of resources to help you get the best value from your sample. If you go to support.10xgenomics.com, you can access and download all of our protocols for free. And if you don't know where to start, we recommend starting with a flowchart entitled Guidelines for Optimal Sample Preparation. It'll help you navigate through all of our available protocols. And our protocols fall into a few general categories. The first category is general sample prep guidelines. So accurate counting of cells and how to isolate cells from nuclei or from suspension cell lines. The second category of protocols we offer are very specific and designed for specific sample types, such as human PBMCs or dissociation of mouse embryonic neural tissue. And finally, the last category of protocols we offer are for general sample improvement, so enrichment of desired cells or depletion of undesired cells. And our 2 current protocols are for enrichment of CD3 positive T cells and removal of dead cells from single-cell suspensions. And both of these protocols use magnetic microbeads from Miltenyi Biotech. And with that, I'll turn it over to Jess.
Hi, everyone. I'd like to give you a few guidelines on general sample preparation tips for running the single-cell platform from 10x. So one of the biggest issues that you face when you're trying to analyze single-cell transcriptomes is that you need a fully dissociated single-cell suspension. Now this is critical because the presence of cellular aggregates or clumps, dead cells or non-cellular nucleic acids or other or potential inhibitors can actually prevent the reverse transcription from working the way we would like it to, and can also prevent you from getting high-quality data. Suspension cell lines, feed-enriched and flow-sorted cells can be used directly after washing. We find that they do not inhibit the reaction in any way. The importance of input cell quality cannot be understated. Ideally, we like cell suspensions to contain greater than 90% viable cells. However, we have had success running this protocol with viability down as low as 30 to 40%. One of the reasons that we suggest protocols like the dead cell removal or removing the fraction of dead cells or non-viable cells is actually the presence of ambient RNA or cellular debris that is present with a high fraction of non-viable cells in your cell population. This can negatively impact quality metrics reported by Cell Ranger. So those of you that are familiar with working with our platform, the metric in particular that I'm referring to is the fraction of reads in cells, which is significantly impacted by the presence of ambient RNA in the system. And cell handling, it's really important to treat cells gently to minimize cell lysis and cell loss. So when cells lyse, obviously the ambient mRNA is released. When you're working at single cell resolution like we are, and we're looking for a single cell being encapsulated in a single partition, having ambient RNA in the system is almost equivalent to running bulk RNA and trying to partition that into the droplets in the system. It basically acts as a contaminant, which again will decrease the fraction of reads in cells, because you're now not looking at single cells and single partitions, you're looking at single cells and single partitions and ambient RNA in the remaining partitions. It's really important to wash your cells twice and use a wide bore pipette during cell preparation. This helps remove contaminants, but it also helps minimize damage that can be inflicted onto the cells during the washing process. And we also recommend to wash cells in PBS plus 0.04% non-acetylated BSA to minimize cell loss during handling and prevent clumping and aggregation of cells. We really strongly recommend debris removal, particularly cell straining, to remove aggregates or debris from washed cells. So there are several reasons why we suggest this. The main one is that the presence of aggregates and debris can actually result in inaccurate counts, either by automatic or manual counting, and I'm referring to things like the count test or the hemocytometer. And really importantly, our gem generation occurs in microfluidic channels that are actually narrower than the width of a human hair. So that's pretty tiny, and the presence of aggregates and debris can actually induce clogging or wetting failures, which we're obviously trying to avoid. Cell counting is incredibly important, and the reason that we have talked about this in many of the sample preparation notes on our website are that approximately 65% of loaded cells will be recovered, and we want to make sure that you can actually obtain that number of recovered cells. So to do so, we recommend operating with input cell concentrations in the range of 700 to 1200 cells per microliter. We recommend loading between 500 to 10,000 or obtaining 500 to 10,000 recovered cells per channel because underloading or overloading may impact your application performance. And we also recommend storage of single cell suspensions on ice after preparation. Now, ideally, we like cells to be loaded into the system within 30 minutes following preparation, but we do understand that this is not always possible, and we have found that keeping them on ice for prolonged periods can actually help maintain the viability of these samples compared to if they were being stored at room temperature. Some more fragile cells may actually decrease in viability significantly if not processed or loaded immediately, and in situations like this, the cell's preferred media can be substituted for the PBS plus BSA combination that we usually suggest. I wanted to take a couple of moments to spotlight a couple of crucial aspects of cell preparation that we've spent a lot of time working on over the last couple of years. And one of these is the importance of gentle pipetting. Everyone knows how to pipette, obviously, and we do this every day in our job, but when we're working with cellular input materials, it actually becomes pretty crucial to treat them gently. We found that rough pipetting cells with a regular bore pipette is equivalent to, or if not worse than, vortexing for 5 seconds. And why is this the case? So what happens is you actually damage the cells, which leads to the presence of ambient RNA in the cell suspension. This lowers the fraction of reads in cells. Like I said before, you get more ambient RNA petitions than you would ordinarily do. And it actually impacts your complexity. So when I say a lower fraction of reads in cells, what I'm referring to is on this typical barcode rank plot, you have a cliff and a knee. You can see this is very steep and sharp. And as you move across the panels from using a wide bore with rough pipetting, a narrow bore with rough pipetting, and vortexing, you can see that the knee or the inflection point between the cliff and the knee actually changes. You get this little wiggle here, and it doesn't become as sharp and as clean as in the control sample. You can also see when you look at the metric fraction of reads in the cells, this decreases as you change the way that you actually handle the cells during the preparation process. You can also see in the library complexity, or the median genes and the median UMIs per cell, that you get a loss in complexity as you start treating the cells differently. And for the purposes of this study, all of the samples were normalized to 50,000 raw reads per cell to make an apples-to-apples comparison. The next spotlight I wanted to place was on washing and resuspension. So washing isolated cells is really important. We typically suggest 2 washes. The speed at which you spin the cells is going to be very dependent on the cell type. For those of you that are familiar with our preparation protocols, you'll know that there are typically 2 speeds that we suggest, and it really depends on cell size. PBMCs are usually run at a slightly higher speed than our human mouse cell mixtures or our cell suspension from cultured cells, and that's mainly due to the size of the cells. We typically always wash, like I said, in a PBS-BSA combination, but if your cells are particularly fragile and susceptible to lysis, you can actually replace that with the cell's favourite media. And like I said before, we particularly suggest using gentle pipette mixing. One of the actually important things is to not invert the tube for accurate cell counting. We've found that when that happens, you obviously tend to lose a lot of cells to the top and the lid of the tube, and that can actually seriously impact your count. And we always recommend adjusting to the desired cell concentration rather than giving you a final resuspension volume. This is important because obviously every cell preparation will differ and the amount of cells going into the washing protocol will be different. So the optimal range here again is to resuspend so you have a concentration of between 700 to 1200 cells per microliter. As I mentioned before, debris removal is really important, and we typically use 2 different types of strainers at 10x. We either use the Flow-Me Cell Strainer, which is a product from Bellart. It's a pipette tip filter. The other option is a Max Smart Strainer from Miltenyi Biotech, and this is a filter that sits on top of tubes, particularly like the conical tube that you can see in the photo. The main aim here is to let the cells pass through while the cellular aggregates and the debris stay on top of the filter. As you can see in the 3 images on the slide, by filtering the cells, we're actually able to remove the cellular debris and the clumps that were present in the unfiltered Jurkat cells. The next spotlight I wanted to place was on the accurate quantitation of input cell suspension. As I mentioned previously, counting cells is really important, and accurately counting cells before loading into the system. So the guidelines that we operate under is our cell suspension optimal for the dynamic range of the counting technique used? We typically use the Countess at 10x, and we like to operate, as I said previously, in the range of 700 to 1200 cells per microliter for 2 reasons. One, we find this gives us the greatest accuracy with our recovered cell loads, but it's also a really good spot for the in the dynamic range of the Countess. We don't want to be over the dynamic range, and we don't want to be under the dynamic range, because that adds a lot of error into your counting. We also perform 3 to 4 reproducible counts per measurement. And we're looking for the standard deviation of these counts to be less than 25%. This is important. And we also try to operate in the range where we pipette between 2.5 to 15 microliters of either our cell or nuclei suspension into the master mix. We find that if we operate at volumes lower than 2.5 microliters, we actually increase the variance in our cell count due to pipetting inaccuracy, while volumes greater than 15 microliters increase the risk of adding in unwanted debris or inhibitors, particularly if filtering isn't an option due to the small volume of cells that you have going in. As I said before, to maximize the likelihood of achieving the desired recovery target, it's crucial to adjust your cell or nuclei suspensions to a concentration of 700 to 1200 cells per microliter. And this can be achieved by actually concentrating your cell suspension. If you find that you have 300 cells per microliter and 500 microliters of PBS-BSA, you can do a gentle spin down and concentrate in half the volume to actually achieve that target. So we have a new flow chart out that describes the guidelines for optimal sample preparation. And this is actually a summary of all the key points that we've mentioned and that are really important going into running any of the single-cell protocols provided by 10x. Some of the key points to consider, and there are actually recommendations provided on the flowchart, is your cell suspension well singulated and free of debris and aggregates? Is your concentration actually within the dynamic range of the counting technique used? Is the standard deviation of these counts less than 25%? Are you in the optimal range of cell concentration, which, like we've said previously, is 700 to 1200 cells per microliter? Is your viability greater than 70%? And have the cells been mixed prior to adding into the master mix? The importance of that step actually can't be understated, because you can spend all of the time prepping the most beautiful cell suspension that follows all of our guidelines, and if you forget to mix it prior to loading into the system and it's been sitting on ice for 30 minutes to an hour, obviously then the chance of actually not hitting your target or having clumpy aggregated cells is a lot higher. So we really strongly suggest mixing the cells prior to loading into the master mix. So Jill and I wanted to take a little bit more time, as she said previously, to talk about 2 of our newly demonstrated protocols, or newly released demonstrated protocols. The first of which is nuclei isolation, which we're excited to actually have a protocol for now. We know this has been a long time in the making. So this is actually a series of 3 protocols that look at isolating nuclei from cultured cell lines, from fresh embryonic mouse neural tissue, and from fresh adult mouse neural tissue. So why nuclei? Nuclei lets you look at cell atlasing without the cells. So some cells just are so fragile that you can't actually work with them, and so actually obtaining the nuclei and running those will give you a better chance at answering the research question you're looking to answer. It expands the application of droplet-based single-cell genomics. So if you have archived samples, damaged or dissociation-resistant tissue samples, you can work through them and get nuclei rather than having a suspension of some intact cells and plenty of damaged cells. It lets you profile complex tissues or organs where nuclei can be isolated, but like I said, intact cells are very hard to come by. It offers the future potential for clinical samples, and it also lets you reveal genetic regulatory mechanisms specific to the nucleus that you may not be able to obtain from the whole cell. So our solution and the protocol that we've put together enables scaling nuclei to 10,000 nuclei per sample, and we've demonstrated that internally that we can actually obtain that many nuclei in a single channel. It's a fairly straightforward protocol that uses all of our pre-existing reagents for the single-cell 3'B2 kit, and it also uses the existing chromium or single-cell instrument. There is no modifications that have been made to either the instrument or the reagent kit. We've found that there's no need to optimize the dropseq protocol that's currently out. There are differences in that protocol to our protocol, and what we've found is that the protocol we've put out is unique for the applications that we're providing. It helps preserve the cleanliness of the sample and helps you obtain the results that you're looking for. It also resolves biological samples differently to single cell. So You still get the same cellular heterogeneity, but you do see things like more intronic and antisense mapping to RNA species. And you also have limited nuclear polyadenylation in. So that does mean that in some cases you will be seeing fewer genes than you would typically expect to see from cells. And this is totally normal. It doesn't mean that the sample's not working. It just means that obviously you're now in the nucleus and they're just simply less polyadenylated genes present. So there are some important considerations when working through our nuclei prep protocol. So the best practices section outlines the fact that this is a protocol that talks about cell lysis, washing, debris removal, counting, and concentrating nuclei. So basically it focuses on all of the things that I've previously mentioned that are important. And again, we focus heavily on minimizing the presence of nuclear aggregate, dead cells going into the protocol, cellular debris, cytoplasmic nucleic acids, and potential inhibitors of reverse transcription. So again, many of the common features about preparing cells actually carry through to nuclei. The sample types that we worked with in this protocol, as I mentioned, were cultured cell lines and embryonic and adult neural tissue. We expect that the protocols we've outlined will be compatible with many, but not all cellular tissue types. Understandably, we can't work with every type of sample, and so we tried to focus on 3 of the major applications and ways that you could work with each of these types of tissues to give you general guidelines going into your particular sample. You may need to add additional optimization steps for cells or tissues that are sensitive to the suspension composition or specific handling techniques. And solid tissues or cryopreserved samples may require additional optimization that we did not cover in the general guidelines presented in our protocol. So we worked with several different cell types and tissues, as I mentioned, suspension cell lines, GERCAT, 293Ts and 3T3 cells, so from humans and mice. We also worked with normal peripheral blood mononuclear cells and the E18 combined cortex, hippocampus, and ventricular zone, and the adult mouse combined cortex, hippocampus, and ventricular zone. And again, just to re-emphasize, the lysis protocol is expected to be compatible with many but not all cell types, and additional optimization may be required for your specific sample type. There are 3 major workflow steps, or there are major workflow steps covered in the 3 protocols that are all fairly similar. The first step to each one of these protocols, whether it's from single-cell suspensions or from tissue, is the actual lysis and nuclei isolation. Now, depending on whether the sample is coming from a cell line or from tissue, this can take between 30 to 45 minutes. The second step in working with tissue samples, so particularly those in protocols 2 and 3, is myelin removal, which takes about 45 minutes. Now, we suggest myelin removal because obviously with samples coming from neural tissue, once you dissociate the actual tissue, myelin is present in copious amounts. And to actually make sure that you get the cleanest results possible, we suggest removing it. In this case, we use myelin removal based from Miltenyi. And in the adult protocol, the third step is density gradient centrifugation, or using a sucrose cushion to actually remove additional cellular debris. As you can imagine, and as some of you probably know, the plasticity of adult tissue is very different from embryonic tissue, and there is a lot more debris that is released upon lysis or dissociation of that tissue. So we want to, again, make sure that your sample is as clean as possible. So that's why we've suggested 2 additional steps. And I'd like to focus on the addition of these 2 steps in the next slide. So why do we suggest myelin removal and density gradient centrifugation? We could just lyse the tissue and we could just run it through the system. So what you can see in the first panel is that this is a countess image taken post-lysis. Pretty gross, to be perfectly honest, and when you actually look at the barcode rank plot, there isn't really a cliff or a knee. It rolls over. The algorithm is having a lot of trouble calling the cells from the background, or in this case, the nuclei from the background, because there is so much debris and ambient RNA present throughout the system. Once we subject the sample to myelin removal, which becomes the second panel, you can see that a lot of debris is removed. One could argue that the concentrations are very different, but we have tried to normalize the concentrations of the actual debris or the cells as as much as possible in these images. So it really has cleaned up the sample. And you can see now that we actually are starting to get a person need. We're starting to have single cell behaviour, which is great. This is exactly what we're looking for to get you the data that you need. Once we add in the additional step of density gradient centrifugation, this person need becomes a lot more defined. You can see that the single cell behaviour is much clearer. It is a lot easier for the algorithm to actually call the cells from the background, or in this case, like I said, the nuclei from the background, which is great. This is exactly what you're looking for. So this is why we suggest going to these additional steps. You can see as well that the numbers and the metrics actually change as you go through these different steps. So again, as I mentioned, the fraction of resin cells is changing. We go from 45% to nearly 60% and then 70% by adding in these additional debris removal steps, which is really important. It means that the sample is becoming cleaner. As you can see that we also get a reduction in the overall mapping rate, as I mentioned before. You get an increase in the level of intronic mapping compared to a traditional cell sample, and the overall mapping to the transcriptome decreases. We've found this is fairly normal with samples that we've worked with in-house. We can still actually do gene expression and clustering, which is the most important thing. And as you can see, when you compare the post-lysis sample with the sample after myelin removal, and after myelin removal with density gradient centrifugation, we can still make similar calls in terms of the gene expression and the actual clustering that's present in the sample. So we can see examples of excitatory neurons and inhibitory neurons. We can see examples of astrocytes being present and oligodendrocytes and radial glial cells. So, as I said, we understand if you don't want to go to the time and effort of doing these additional steps, but from our experience, the data or the quality of the data, it becomes significantly better if you do invest the time in actually adding in these debris removal steps. Additional points to consider as you're going through this protocol. As I said, there are a lot of areas that can be optimized, particularly because the tissue and sample types that you as our customers might want to work with are very different than what we have access to in-house. So one of the things that we stress the importance of is the lysis time. It's really important to monitor lysis efficacy to determine optimal conditions. Once you work through this protocol on your cell type, you'll know the amount of time taken to lyse the cells or the tissue adequately. If it's the first time that you're working with the protocol, we suggest that you monitor after 3 minutes on a cell suspension and after 15 minutes on tissue. So again, checking the viability of the sample via imaging techniques, either microscopy or via the counter cell hemocytometer is important. You may be thinking, how can we monitor the viability of nuclei because they appear dead? In this case, we're actually suggesting that you look for the presence of live cells. You want the fraction of live cells in your sample to be less than 5%, so that the signal that you're getting is coming from the nuclei rather than having a high abundance of sample cells. We also suggest monitoring lysis time because we want to avoid over-lysing cells. This may actually mean that you're lysing your nuclei, and again, you will see more RNA transcripts and you're not going to get the library complexity that you're looking for. Each cell type and tissue type will be slightly different, which is why we place emphasis on monitoring the lysis time. If you find that your input cells and tissue are sticky or that the nuclei are starting to aggregate easily, you can increase the concentration of BSA in your suspension from 1% to 2%. This may help create a nice single cell suspension or a single nuclei suspension. Centrifugation speeds and times are important, and again, it's important to optimize these to minimize the loss of nuclei as you're going through the washing protocol. When working with a new sample type, and this is very similar both for cells and nuclei, we recommend saving and counting the nuclei of the cells in the supernatant to ensure that the losses are actually minimal. If you find that you're getting more cells present in your supernatant than are in your final suspension, that's a good indication that you need to adjust the centrifugation speed and time. And for density gradient centrifugation, we've used a particular sucrose gradient. You may find that there's sucrose gradient conditions that work better for you. We've also done this on a benchtop centrifuge rather than a swinging bucket centrifuge. So again, that can be adjusted. And you may also prefer to try an 10x-pert if that's your preferred method of sucrose gradient. Things that we do not recommend are mechanical disruption of cellular membranes, particularly things like down homogenization. What we have found is that it is very difficult to control for the time and pressure between users and different cells or tissue types. We do not recommend reducing the concentration of BSA in the buffer because this will result in nuclei aggregation. We do not recommend replacing the nuclei wash and resuspension buffer with PBS plus 0.04% BSA. What does tend to happen in this case is that the nuclei irreversibly clump into a ball and you can't actually pipette them properly. So you basically end up loading PBS into your reaction mixture without getting any nuclei, which is obviously a worst case scenario. We do not recommend operating at room temperature, so we found that keeping the nuclei cold during both lysis and centrifugation actually really helps with the integrity of the sample. We do not recommend using improperly stored tissues, and by this I mean samples that are stored, for example, at 4 degrees for periods of time, longer than recommended either by the provider, if you're working with a service provider, or outside your normal storage conditions. And we do not recommend performing nuclei isolation on mixed species simultaneously. So what I mean here is using barnyard cells, which is the classical 293T 3T3 combination, to assess system performance. If you take this sample type and you run the nuclear isolation protocol on the mixed samples, what we find is the nuclei tend to aggregate together during the protocol. If you actually take the cell lines and lyse them separately and then mix them post-lysis, we find that they actually don't aggregate and we can obtain multiplet rates equivalent to our single-cell suspension. We've tested alternative washing and resuspension buffers in-house instead of the buffer that we recommend. I've listed them on the slide here. What we've found is using any of these results in significant aggregation of the nuclei, which seriously impacts the quality of your data. So we do not recommend substituting any of these for the buffer that we've recommended in the protocol. I quickly wanted to highlight some results that we've obtained. So looking at gene expression between replicates of nuclei isolated from adult mouse neuronal tissue. So we ran a P6 mouse combined cortex, hippocampus and ventricular zones in according to the guidelines in Protocol 3, we ran replicates of the sample. As you can see from the cell loop profiles, they actually overlay perfectly. And when you look at the correlation in terms of the average gene expression between the two samples, they're very strongly correlated, which is great. When we perform a similar experiment, but this time we look at two separate embryonic brain sections, so cortex, hippocampus, and ventricular zone. One was dissociated to obtain cells according to our mouse embryonic neural tissue protocol. The other was actually lysed to obtain nuclei directly from tissue. And we're looking at the correlations now between the cells and the tissue. You can see that the correlation is strong. We're looking at the correlation between replicates of the cells, which again is slightly stronger correlation, and again the replicate of the nuclei isolated from the tissue, and again the correlation is strong here. So we are seeing, what we're seeing is really nice actually, that we can obtain similar levels or similar types of gene expression markers from cells and nuclei from embryonic tissue. When you look at this using Leaf Cell Browser and you look at the neuronal cells in the purple and blue boxes and the nuclei in the pink and yellow, you can see again very, very similar profiles. Slight differences, particularly with little groups of clusters here, but they look very, very similar. And again, you can go through and you can run differential gene expression on the cells and the tissue types. And you can, again, see the same types of things. You can see excitatory neurons, you can see inhibitory neurons, you can see astrocytes, oligodendrocytes, and radioglial cells, which is really nice. So now I'm going to hand it over to Jill, who's actually going to place a spotlight on our other new demonstrated protocol of dead cell removal.
Thanks, Jess. So as Jess mentioned, we have a new protocol available on our website, and it's a general sample improvement method for removing dead or non-viable cells from a single-cell suspension. Now, the protocol for removing dead cells is pretty simple. I won't run you through the details to save time, but essentially you resuspend your cell pellet with the dead cell removal microbeads, run them through a magnetic column, and the dead and non-viable cells, which were tagged with the beads, will stay bound to the column, and the effluent is going to contain your viable cells. So you just take that effluent, wash and resuspend, and you'll be ready to run an R3' assay. The images on the right show sort of the before and after, and you can notice the dramatic improvement in viable cell percentage after the microbeads, and that's the image on the bottom. Now, to test this technique, we did an experiment using human PBMCs, and we have 4 different treatment conditions. One was a control treatment where we loaded cells immediately for partitioning after sample prep. For the next 3 samples, we had 3 different ways to induce non-viable cells. So the first was just to leave the suspension of PBMCs out for 24 hours at room temperature, and that reduced the viability to around 50%. The next method is to treat cells with digitonin, so you end up with a very non-viable cell population. And we mix the non-viable cells with viable cells to end up with about a 1-to-1 ratio, so 50% live, 50% non-viable. And then the last condition was a heavy digitonin treatment, where we took the same digitonin-treated cells, mixed them with viable cells at a 5-to-1 ratio, so we ended up with around only 20% viable cells. And as you can see in the data, the top panel, these are all of the pre-dead cell removal conditions, and at the bottom are the post-dead cell removal conditions. And the thing that's probably going to jump out to you is that on the bottom, post-dead cell removal, all of the barcode rank plots look essentially the same across all 4 conditions. Whereas if you look at the barcode rank plots for before dead cell removal, you can see the control cells look fine. 24 hours at room temperature don't look too bad, but once you get into the digitonin treatment, barcode rank plots seem to indicate significant quality issues with the sample. Some of the metrics that you'll see improve: viability on the top, 85%, 50%, 50%, 20%. After dead cell removal, you're in the 80% for all of these conditions. And again, this protocol is pretty quick, so it's a small amount of time to really get a dramatic increase in cell viability. One of the most important metrics for sample quality is fraction of reads in cells. And you can see before dead cell removal, you're getting down to 50 and 60, in the 60% for fraction of reads in cells. But after cleanup with the dead cell removal beads, you're back up to high 80s and 90s. Some additional metrics that will improve are the number of median genes per cell. You can see at the top our control condition. This is at 50,000 raw reads per cell. You're starting with close to 1,000 genes detected per cell. And then for the most extreme digitonin treatment, you're down to only 27 genes detected per cell, which is definitely indicative of a low-quality sample. But in addition to the system metrics, let's look at what happens from an application standpoint in cell clustering, pre- and post-dead cell removal. So this is the control condition, and we're looking for T cells, cytotoxic CD8 T cells, B cells, and monocytes. And before dead cell removal, after dead cell removal, you can see the identification of cell clusters is essentially the same. And this is what we're expecting to see. And it also indicates that the dead cell removal beads don't have a negative impact on differential gene expression or clustering. If we switch to the samples that were left for 24 hours at room temp, you can see that the clustering profiles are different overall. And this might be biological due to the fact that the sample sat overnight at room temperature. And you can still call the clusters before and after dead cell removal. However, if we go to the digitonin cell mixtures, you can see on the top, clustering performance is significantly reduced. But after dead cell removal, we're left with clustering that looks essentially like our control sample. And this is even more dramatic for the sample that started out at 20% viability. After the dead cell removal protocol, you end up with library samples, differential gene expression that looks essentially like our control untreated cells. So this is a really powerful way to boost application performance. One of the main reasons that this protocol works effectively is that this reduces the amount of ambient RNA that comes from dead or dying cells. And as just mentioned earlier, the cell ranger metric called fraction of reads in cells really reports on how much ambient RNA is present in the system. When you clean up the suspension, when you get rid of the dead and dying cells, that metric improves and leads to other improvements such as median genes per cell and better cell clustering. But what's important to note is that although the endpoint was the same for all four of these experimental conditions, the delta, or the degree of improvement, is really going to depend on the quality of the starting material. Samples that start out better will not see as dramatic of an improvement as samples that start out with in a much poorer state. So that delta is going to depend on what you're working with. But as a general guideline, we recommend dead cell removal if you have a suspension with around 70% or less viable cells. But of course, you can tune that to whatever your particular sample type or application goals are. And so just to summarize, we walked you through just two of our protocols today, the isolation of nuclei and removal of dead cells. We have many more resources available online. And again, if you're just getting started with single-cell sequencing, download the flowchart guidelines for optimal sample preparation, and that'll really put you on the right path. So thank you very much for your attention. And Shauna, if you'd like to start feeding us questions, we can do a little Q&A.
Sure, thanks, Jill. Thanks for a great presentation to Jill and Jess. So we're going to get started with our Q&A session. Remember that if you have a question for our 10 experts, please type it in the Q&A box that's on the right side of your screen, and we're going to do our best to get through as many questions as we can. And I'm going to do them in order that they came in. So, all right, let's get started. So our first question is, could you address single-cell isolation from microdissected brain tissue?
Okay, so we've never actually worked with that tissue type. It's exciting that you would like to work with that tissue type. I wish we could do that here. There's no reason why, theoretically, you couldn't obtain a single-cell suspension from this tissue type. We are currently working on protocols that focus on better ways to dissociate brain tissues, and they should be out, hopefully, within the next 6 to 12 months. But using the general guidance that we've provided on some of our mouse protocols should work for you. Additionally, you could also try the nuclear preparation protocol if you find that actually obtaining cells from that dissected tissue is damaging the actual cells and you can't obtain a single cell suspension. Nuclei may possibly be the way to go there.
Okay, thanks, Jess. And we'll go on to the next question. In internal testing or through customer feedback and assuming proper sample handling, have you seen any cell types that capture better or worse than others?
That's actually a really good question. Internally, no, we haven't. We try and work with as many different cell types, particularly different cell sizes as possible, to see whether there's actually some kind of encapsulation bias. We personally haven't seen that in-house, but again, we typically try and operate very, very closely to the guidelines that I presented earlier. So the importance of cell counts and getting the concentrations as close as possible into that 700 to 1200 range, we find that we actually are usually really on target with our cell encapsulation and our recovery. From a customer standpoint, I would suggest that if you notice that you are having problems or you think you're having problems with cell encapsulation or certain cells are capturing better than others, I guess my first question would always be, were the counts for those types of cells very similar? And if they weren't, if you can normalize those cell counts, I think that you will definitely help in that case capturing cells better or not seeing bias.
All right, we'll head on to the next question. So this is in reference to washing, preparing the single cells. When you were doing the presentation, can you better define what you mean by spin down when you're talking about that protocol? Is it talking in reference to using a centrifuge?
Yes, absolutely. So every time we reference spin down cells, we're always using a centrifuge. And depending on the number of cells, it's either a small-scale benchtop centrifuge or it's a much larger centrifuge. we do this obviously because we want to be able to pellet the cells in the best way that we can, so it's always in reference to centrifugation. Like I said, the spin speed and time is going to be highly dependent on the cell type that we're using. If you look at the different demonstrated protocols that we have on offer on the website, you'll notice that speeds vary from 150 RCF up to 500 RCF, depending on the protocol and the type of cell or nuclei that you're actually isolating.
And you can check to see if you're spinning effectively.
Yes.
You can look at the soup and see if you have cells or nuclei left over. And that's, as Jess mentioned earlier, it's a good technique for sort of getting right into the sweet spot where you need to be, because every centrifuge operates a little bit differently. So 300 RCF on our centrifuge might not be exactly equivalent on yours, but there are really nice empirical ways that you can check that.
Yeah.
All right, and the next question. So how long do we have between gem preparation and RT reaction? Sometime after the gem generation, there is like two phases that appear in the same sample by the time that they start the RT reaction. Is it detrimental? How do you avoid this issue?
Okay, so I think I know what you're talking about, and please feel free to ask a follow-up question if I don't answer this to your satisfaction. So the two phases that I believe you're talking about are the separation of the partitioning oil from the actual gem. This is completely normal and is to be expected. If you were to pull the PCR plate or the strip tube out of the thermal cycler after the RT reaction, you would notice the same phenotype, that you have a separation of layers. The partitioning oil always sinks to the bottom, and the gems are always at the top, just based on density. Completely normal. We have tested up to 2 hours with the cells actually being partitioned in gems before you actually load them into the thermal cycler. We obviously don't recommend this if you only have 4 samples, but we've gone through and prepared samples back to back to actually look at running multiple chips in a row. How long can you actually leave the cells there? And everything's fine. Like, as long as the partition gems are in a plate or a strip tube that is on ice, it's completely normal, and you should notice no ill effects in your data from the apparent separation of these two phases. Like I said, it's completely normal.
All right, thanks. So in reference to the nuclei protocol, would the myelin removal step be necessary for a tissue other than the brain?
No. That step in particular is particularly relevant for brain tissue because there is so much myelin present. Obviously, different types of tissues will not have myelin, or potentially will have different degrees of myelin. So no, if you're working with a different tissue type, there is a very, very good chance that you definitely don't need that, and that maybe all you might require is a sucrose gradient step if you find that there is a lot of debris present. I would suggest that for tissue, there will be a lot of debris present. For cell samples, in our experience, we've found that there isn't a lot of debris, which is why we can just slice the cells, wash, and then go straight into the protocol, or the single cell protocol.
All right. So on the slide that suggested using OptiPrep in the nuclei protocol, what about using OptiPrep in the regular single-cell protocol? And just to clarify this, they were curious about this because the use of the OptiPrep is used in the InDrop platform for their single-cell sample prep.
Yeah, so we've definitely had customers that have used OptiPrep on neural preparations. They find that helps them clean out the debris and get much cleaner results, and they seem to be very happy with that. So yes, absolutely, please feel free, if you see that your sample contains a lot of debris and you're very comfortable with using that, please feel free to use it and run samples through the system. Like I said, we've had reports that it works very well.
Yeah, and along those lines, there are other ways to remove debris from solution as well, like using a flow sorter. Yeah. There are a lot of methods that our customers use that work just fine.
Yeah.
All right. So the next question, when you want to do single-cell RNA-seq, we're not focused on the study of any nuclear mechanisms, but just want to recover the most transcripts. Do you recommend doing the nuclei isolation?
You want to focus on getting back the most transcripts. If that was the case, I would always say focus on cells. Cells are always going to contain a higher number of transcripts than nuclei. If you can't actually obtain intact cells, then there's definitely the way to go in that case. But yeah, if you want to obtain the most transcripts, whole cells are definitely the preferred method, yeah.
Great, thanks. And so these two questions, they came in from different people, but they're in reference to the dead cell removal protocol section. On the slides with the data, how did you normalize the mean reads per cell? And the second question is, how was the cell viability estimated?
The cell viability is estimated from measurements on the campus. That's, as we mentioned before, typically our method of choice in-house. So we're doing just trypan blue staining and looking at what the campus is telling us is live and dead.
And median to high 80s is typical of what we see on the campus for looking at PBMC viability.
Yeah.
It's not 100%.
And then normalizing read depth, what we do is we actually we basically run downsampling, which you can do, or you can also use Cell Ranger Ag, and that basically lets you aggregate all of the samples, which in this case we have aggregated all of them to look at the gene expression, and you can specify a level that you want to downsample to, or a number of reads per cell that you want to downsample to. In most cases, we typically choose to downsample to 50,000 raw reads per cell because this gives you a fairly good indication of the level of complexity of your samples. But like I said, Cell Ranger Ag, or the aggregation pipeline will actually let you do this and specify that downsampling metric.
All right. And the next one, we are at our facility not currently have wide bore chips. They are not included in the sample prep protocol. But do you recommend that we use a wide bore chip for the cell resuspension and loading into the cartridge?
So the general guidance is when you're preparing cells, and so if you're in a tissue culture room or wherever it is you actually prepare your single cell suspension, always use a wide bore pipette. That just helps generally in terms of your sample quality. Once the cells actually leave the tissue culture room or where you're actually preparing them and you're about to load your chip, you can use a regular bore pipette to actually mix the cells and actually load them into the system. That seems to work perfectly fine. It's just those initial first steps once you change the media and move into PBS that we find are the most sensitive to sample handling techniques. And so wide bore pipette is always strongly recommended compared to a narrow bore or a full bore technique. If you find that your samples are very, very clumpy to begin with and contain a lot of aggregates, in that case it is suggested to use a narrow bore pipette that may help break up those clumps on the first round of washing and resuspension, but it's rare that that happens.
And the next question, for the pre and post dead cell removal, how many cells were you targeting?
In each case, roughly 1,200 cells was targeted in pre and post dead cell removal.
Okay. Next question. What types of volumes do you recommend for the dead cell removal? For example, could you utilize this protocol with extremely limited samples?
It becomes more tricky when you have extremely limited samples. So if you have less than 100,000 cells to begin with, we do have a section in our general cell preparation protocol that talks about simply concentrating and running the cell sample as is. I've certainly done that internally when I don't have a lot of cells and I know that my viability isn't good, but I need the data, so I'm just going to go ahead and do that anyway. We typically, in this case, have worked with like 1E6 to, about 1E6 cells going into the protocol. If you have less than that, it may be trickier to actually get this to work. In that case, I would probably say there may be some need to actually consult Miltenyi on whether there is better ways to actually deal with limited cells. Maybe there is volumes of buffers or feeds that might need to change. But like I said, we typically work with 1E6 cells going into this protocol.
All right. Next question. You mentioned that the channel width of the chip is less than 100 microns. Is this the same size for the V2 chemistry as V1, or is the V1 chip channel narrower?
No, they are the same size, yeah.
Okay. So I'm working with cells that are smaller than normal rodent neuronal cells. I'm wondering what you would recommend on sample preparation to avoid doublets.
Okay, so we haven't seen an elevated doublet rate based on cell size. The way that the microfluidics works and the way that the cells are loaded into each gem is by limiting dilution, and so it's not a function of having, cell size doesn't seem to play a huge role here. So I would suggest that you simply treat the cells as you normally would, regardless of the size, and I would load the same number of cells that you would consider doing in a normal experiment. You shouldn't see that you have a higher multiplet rate because the cells are small. Like I mentioned with our nuclear protocol, obviously the nuclei are a lot smaller than cells, and we typically don't see a higher multiplet rate than we'd expect if we prepare them according to the guidelines that I outlined. And again, like I said, they are very, very small in this case.
All right. And then this is just a similar question. Do we know or have any experience with the doublet rate for nuclei? Is it similar to our single cell?
Yeah, so like I said, it's, when we lyse the human cells and the mouse cells separately and then we combine them post-lysis, yes, the multiplet rate appears to be actually really consistent with our cells, which, to be perfectly honest, surprised me because I wasn't expecting it to look so good. And we tested that over the dynamic range of the assay, so from 500 up to 10,000, we made sure that we looked at that. What we did find, obviously, as I also said, was if we lysed them while the cells were actually combined together, we found that they stuck a lot more, and so that the multiplet rate was exaggerated, a lot higher than we'd expect it to see. So that's why my guidance would be to, if you want to do mixed species experiments, isolate your cells separately, isolate your nuclei separately and then combine them. And you should be seeing multiplet rates that are consistent with a single cell suspension of the same species.
All right. What about the heterologous cell size tissue, like adipose tissue? Is there a good strategy?
We haven't worked with adipose tissue ourselves. I know that some of our customers have worked with adipose tissue and have had success. Was the question about isolating cells from tissue, or more just about does the tissue or those cells actually work in the system? If it's a question about the cells working in the system, like you said, we've had some customers that have worked with it. I'm sure we can provide some general guidance if it's a question about isolating the cells from tissue. Please feel free to email support@10x-genomics. Your questions actually do come through to R&D. We do answer them for you, so please feel free to reach out to support if there are specific issues that you'd also like to talk about as a result of today's Q&A.
All right. So are you working on a dissociation protocol for human tumor tissue?
Yes, we are. We're developing that internally, so it'll be a while before it reaches a customer-facing document, but it's definitely something we're working on. Great. And if there are particular tumor types you're interested in, write to support.10x and let us know, because it's a wide search space, so we want to make sure that we focus on the tissue types that are most relevant for our customers. So we do really enjoy feedback and suggestions. Yes.
All right, and I think there's time for one last question, and I know there are a lot more questions in there, and we will be able to address those as well through the community. So the last question for today is, if using a hemocytometer instead of the Countess for cell counting, will the cell strainer step alter the final cell count, since there might be volume loss during the strainer step?
Well, honestly, it should be the same. I mean, if you strain your cells pre-counting, regardless of whether it's a manual or automated counter, you should ideally be obtaining the same count, regardless of, again, whether it's automated or manual. I would strongly suggest, if you're actually really concerned about the count pre-filtering and then post-filtering, and then you can actually determine loss. So we always suggest to count post-filtering because, again, there can be losses regardless of the counting technique.
Okay, so that about wraps us up for today. I just want to thank our 10 experts, Jill and Jeff, and our attendees for your questions. We will be posting a recording of this session on the 10x community shortly. So if you missed any part of it or you want to review it, it will be available on the community. And if we didn't get to your question, or if you have a follow-up question, you can post it on the community in our single-cell sample prep forum at community.10xgenomics.com, or you can also contact support at 10xgenomics.com as well. And also, we'd love your feedback about what you liked about the session, what you didn't like, any topics you'd like to see in the future. So we invite you to fill out a brief survey right at the end of this session. And thank you very much for your time, and have a great rest of your day.